right? So got it.
Yeah, speaker should come out with that.
Okay, cool. Okay, so maybe we should get a confirmation from someone. Can someone in the call just let me know if you can hear me, if you can see the slides? Okay, awesome. Thank you, Ming. Cool. Okay, so what do I do with this? I'll toss this somewhere. Okay, so hello to in person and hello to online. So I'm going to be talking about this tool called Crescent, mainly going to cover single cell RNA sequencing and analysis, how to get data into the system. Crescent is essentially an acronym, as all scientists love acronyms, Cancer Single Cell Expression Toolkit. So this is a portal that we built around like 2018 to 2020, 2021. And essentially, it's just a tool that will make biologists' lives easier, hopefully. So the idea is to bring your data and have an early exploration into it, specifically single-cell RNA-seq data, and provide sort of tools and high-performance compute in order to do these analyses. So usually with single-cell analyses, we have the need of high-performance compute. So certain labs that are generating this data might not have access to that compute. There's also the fact that single-cell analyses requires complex workflows and expertise involved to tailor those workflows, and usually happens in programming languages like Python and R, where biologists might not be familiar or might be familiar, but might be a little bit intimidating. So the idea around Crescent is to streamline all of that, turn all these very specific workflows and pipelines into a series of buttons and a web app. So what are we going to be talking about today? The motivation behind Crescent, and why do we care about single cell? Why do we care about creating this streamlined analysis and standardized workflows? So once we start talking about that, I'll introduce Crescent, do a little overview, talk about some of the pipelines that we have and some data exploration. But mainly, we're going to jump into a demo today. We're going to use Crescent. I'm going to provide you data. You're going to run it. We're going to see how to kind of play around with Crescent, get an early glimpse into your single-cell RNA-seq data. So the motivation. So the tumor is a complex microenvironment that consists of many different cells. A tumor is not just cancer cells. There are all kinds of floating immune cells. We can have a set of normal cells. A complex environment that needs to be sort of abstracted in order to actually treat the tumor or the patient themselves. So something that we can do about this heterogeneity and to delineate this heterogeneity is this cool new lab tool called single-cell RNA-seq. So similar to RNA sequencing, we get the gene expression of cells through mRNA transcripts. And something neat about single cell in these recent times, there's a whole bunch of these different techniques that you can do. And one of the most popular ones, and the reason why, one of the reasons why single cell sequencing became so cheap was with 10x chromium. So with this fluid, microfluidics based encapsulation system, we're able to separate cells into their these little beads of oil and run the the analysis or the different experiments within each cell or within each bubble at least. And so there's been an explosion of single cell data growth. So as the years go by, there's more and more studies being published. And I think in like 2020, it felt like every week there was a new Nature paper that was saying we discovered this new cell, and it was really exciting. The same type of growth is seen today, but definitely into different modalities of single cell. So we have single cell genomics now, single cell spatial transcriptomics, all kinds of different types of modalities like SC-ATAC-seq and so on and so forth. And what's interesting here in this article or the journal from Svensson was, there's this explosion of growth, but we can see that as analyses have more and more cells, we're determining that there are more and more clusters or cell types because of this. And so what this means is as our sequencing output grows, and as we sequence more and more cells and in a cheaper and faster and more streamlined way, we're going to be able to find more and more cell types. And so with data comes a lot of tools to analyze those data, said data. So we have all these bioconductor packages, all these R tools. We have this little web app called scRNA Tools, not by us, but it's a good tool to check, what's the current date. So this was as of last year. So this is already, updated, but it shows that there's a vast amount of categories of tools from visualization, network analysis, alignment, variance, all that kind of stuff. So there's all kinds of action happening in this space. There's all kinds of data. So with this explosion of data and these explosion of tools, how do we manage these as researchers? How do we try to actually tackle this bigger problem of single cell RNA-seq analysis? And that's when we introduced Crescent. So Crescent is like our one-stop, I don't even know if that's how it goes, but one-stop solution. So essentially you bring your single cell RNA-seq data. Usually this is in the format of gene by cell matrices or MTX data, usually coming out of 10x Chromium and post-alignment. You can also bring in some cell metadata. So if you have very specific, cell cultures or labels for these cells, then you can bring those in as well. And Crescent provides a whole bunch of reference data sets. So this kind of, effort is ongoing, but currently there's about 300,000, public or 300,000 cells covering 20 projects of published data. And the entirety of Crescent is holding currently over 150 million cells across all kinds of private and public projects. And I'll talk about that later, about that kind of discrepancy between private and public. So once you have all this stuff, you can kind of tap into Crescent, and this is accessible via the web. It's just crescent.cloud on your web browser. And what we do is we provide these analyses and kind of the way that we're doing it, I will talk about in a second, but essentially a bunch of magic happens, and then, you get all these really cool interactive plots. Obviously it's not magic. There's obviously, rhyme to this, the madness, or that's not how that goes either. I'm really off today, but rhyme or reason, is that what it is? So there literally is a reason and a way that we're handling all of these. And I'll talk about that more, about what tools we're using, how we're able to kind of package up all these tools into essentially buttons. And, once you get your interactive analyses, you can start to explore your data, and all of this happens inside of the web app. So this work was published in 2020 in Nucleic Acids Research. That was our initial paper talking about a single data set analysis using Crescent. And stemming from that, there's been a couple of interesting papers, one from one of our lab members who's graduated, or a PhD student who's graduated. And, she looked at different injury response states affecting glioblastoma, and how the well, with glioblastoma you have this heterogeneity of cells and clonal different types of clones that might distract from possible treatment options. And so she was able to kind of delineate that these cells came from developmental and injury response networks, which was really interesting. What we did for Crescent for that was we put our data up in Crescent, we made it publicly available. You can also explore the data the same way she did, and a few other kind of cool things about that. There was also this paper from Oliva and Lillian's group, and essentially they split mutants. Oops, sorry, Catherine. Oh, I can't mute. I don't have mute capabilities. Okay, so basically they looked at two different treatment groups. One, a combination therapy, this is for oral cavity cancer, and one, single treatment option. And they wanted to make this this immunotherapy kind of therapeutic call, whether the combination therapy would be more effective. And so what we did with Crescent was we analyzed the patient's blood and immune system and all kinds of different cells in order to see whether the immunotherapy actually changed the results. So in essence, we should be seeing a larger infiltration of immune cells based on this immunotherapy. And so the main work from Crescent was the postdoc at our lab previously, Javier, and Hugh was the one that spearheaded that. So that was really cool as well. And then we have this pre-publication or bioRxiv paper that we tried to compare a bunch of different data integration methods, which is something I'll talk about later about anchoring and data integration. And we tried to replicate the exact results that Laura found in her work, and also tried to make visualizations that represented almost comparative publication-like visualizations. So normally you would stack these plots against each other in R, and instead we're trying to kind of show that all in one web app. So that's some of the things that Crescent has done. There's a bunch of other papers that if you want to go snoop through the NAR paper. But how did we do it? So what's happening in this this little box here? So what's happening is we have these single cell workflow scripts. I have it here as Surat, and Surat is one of the most well-known and well-maintained single cell analyses toolkits from Satija Lab. There's a few other tools that we include as well. There's some custom QC, there's some GSVA, and all kinds of other tools that we're now starting to add on. But the basis of it is a Surat single cell workflow via R scripts. And what we're doing is we're basically wrapping each one of these R scripts into a thing called CWL. And CWL just makes it easy to map inputs and outputs. I think definitely Kobe was really good for this, for mapping out all the different workflow steps. But what we're doing is essentially we're building these files that actually do run, and each step has its own set of packages that it pulls in, and you pass the data in, it runs the packages, spits out the output, so on and so forth. We're also using this thing from GH that enables us to run all of these pipelines. So just a simple, how do we get all this done? And so, talking about, how it was done, we need to talk about also what's in these R scripts. So the current single cell RNA-seq pipeline or analysis pipeline goes through a series of steps, usually about 9 or 10 steps. And it starts at mapping. This is not something that Crescent currently does. Usually what happens is from the lab, you'll get your gene expression data and your single cell data usually mapped straight from the facility. And all you're really giving them is like which reference genome that you want or reference transcriptome. So that part is kind of handled for us. If it's not, then you're going to have to look into something called Cell Ranger if you're using 10x or whatever, whatever recommended mapping tool. And the way mapping works is all these, RNA reads are transcribed, and we look at the DNA, but we map it back as chunks into the human transcriptome. So when that does happen, the difference between RNA-seq mapping and single-cell RNA-seq mapping is that you need to have splice-aware mapping as well as barcode-aware. So it's a slightly different process than RNA-seq mapping, but there's all these really well-maintained and sophisticated tools for these things. So Crescent doesn't handle that because FASTQ files are quite large. Handling that over web app is not ideal. But what comes out of Cell Ranger and mapping is very interesting because essentially you just get a gene by cell matrix, and we can, those matrices usually have a lot of zeros, so we can condense that matrix down and upload a really small zip file into Crescent. So the first step that Crescent kind of takes care of is QC. So you will kind of set your different QC parameters. This is the time where you want to filter out for certain cells, such as based on the counts of RNA or the counts of genes or the mitochondrial count, which might tell you the health of the cell. So dying cell, not very useful in single cell RNA-seq. Also, cells that have been trapped into two cells that have been trapped into a bubble will show way more transcripts, and those have to be filtered out. We call them doublets. So a lot of this QC is kind of handled by Crescent, but you can change all of these parameters yourself as well. Normalization. So this is important when you have different sequencing depths. So you might have cells that have just generally more reads because maybe they're bigger. And so you need to normalize for things like that. Batch correction, this step is important when especially for integration, when you have different modalities, but usually batch correction we do because there might be technical artifacts because the experiments themselves, or the experiments might have been done by different times, at different times, or different people, and or totally different types of data themselves, right? Dimension reduction, we have a huge matrix of hundreds of thousands or tens of thousands of cells with 3,000 or 30 up to 30,000 genes. So you have this huge matrix that you need to kind of reduce with PCA in order to actually have some kind of valid visualizations, which we will kind of do with cell clustering and UMAPs and TSNEs and all that kind of stuff. So I'm not going to dive too deep into kind of like what is UMAP and stuff. I'll talk about the differences between UMAP and TSNE, but for the most part, it's just a dimensionality reduction done so that our human brains can compute two dimensions of data versus the many millions of dimensions of data, right? And then finally, well, we can do differential gene expression. So once we have these cell clusters, once we've separated these cells into their different colors and clusters and what we kind of see as these like TSNE maps, we can also do the differential gene expression. So what genes are actually significantly expressed compared to another cluster, and that might tell you, this group of cells is a clone, and they express quite similarly, except there's a different gene expression that's being expressed in this very specific cluster, and so means more exploration needed type of thing, right? And this isn't doesn't have to be linear. It can be, and in Crescent it is, but this process can you can go back and forth between visualization. Maybe your QC needs to be updated. If you have data like single cell nuclei seq, you won't have any mitochondrial cells, so you don't filter by mitochondrial, and that's not a very good way to see whether the cell's dying now because it's all coming from the nucleus, so on and so forth. There's a lot of these back and forth that you can do, and Crescent allows you to do that, though it does kind of try to fit you into this pipeline so that you get your results back faster. The last thing I've mentioned was cell cluster labeling. We use GSVA based on some methods, some benchmark studies, but there are probably different methods available now. GSVA mainly used for pathway analysis, but what we do is we kind of average out the clusters in order to do like a 30-second kind of cluster labeling. So we average out the clusters. It kind of turns into like a bulk RNA-seq type of labeling where we label the entire cluster. So there's also one other part that I haven't mentioned here. It's kind of experimental right now, but in first CNV, it does copy number analysis. Of course, these aren't DNA, so we can't really take the copy number at face value, but it's pretty interesting to see some of those results as well. If I have some time, I'll cover that as well. Okay, so with all that said, now we have data exploration. So this is what we're going to kind of dive into today. We have a bunch of different visualizations available to us, a UMAP or a TISNI, and we can get our cell clusters that were generated by the pipelines themselves. And we can filter our metadata. So if we have our own cell labels, for example, here, this data is coming from Nature Communications paper, and the author data, they had labeled cells. So, we are able to visualize them as the authors wanted them labeled, or, if you're bringing in your own data, then you might have your own kind of self-filtering that happens experimentally. We can also have all kinds of different labels like, BCR receptor, TCR receptor, all these kind of different mappings that have been done experimentally can be just brought in as text files and uploaded and mapped to this existing visualization. Another really important thing that Crescent brings is interactive gene expression exploration. So you can see this little B and C at the top here. So we're looking at this very specific gene CMC1, and you can see that, most likely it's relevant to this cluster 3. If CMC1 was representative of a very specific gene, or maybe an unknown cancer type or cell, then it would be kind of interesting to see that, only this cluster expresses, CMC1, right? And we can look at other types of numeric metadata as well. So if you brought in, numeric metadata because of let's say mitochondrial counts or something like that, then that can be visualized as well. Okay, so before I start with the demo, is there any questions on kind of the intro there? Okay, I guess I'll give it a second. Okay, so how we can get started. So the first thing that you want to do is you probably want to head to crescent.cloud, and that's pretty much all you have to type is Crescent, and Crescent is spelled like crescent moon, and .cloud. So in your web browser, if you head over to crescent.cloud, the very first part that you're going to see, and the very first kind of thing that you want to check out is you have this, home or main menu tab at the top. So this button here just takes you back to this page, which is the home. You can send us some bug reports, but the kind of main thing that I want you to focus on the taskbar is you have this info, and I'll post up a slide with all the resources. I'll provide the slides, but this info icon kind of gives you all the resources that you need. So there's a couple of things here that are kind of relevant. So once we finish this demo, maybe you didn't catch a certain part of this, you can go check out our documentation. It's linked here, and it will tell you kind of with steps and videos and all that cool stuff. So you're not going to miss anything. There's also our GitHub if you want to check out any of our work, or it's all open source, so that makes it easy to kind of actually check up on us. If you want to email us for any reason. Obviously, we can also contact via Slack and the Panoramic Slack. And also, the NAR paper here is linked as well. So the last thing here on the menu is your user. So when you're logged in, what you'll see is, actually, that's not going to show, but what you'll see is public user. And so what are the constraints of a public user? Well, it's stored based on your cookie. So the second, your browser refreshes your cookie for some reason, you're going to lose all that stuff that you put there. So what I would recommend is to, register. You just put in an email and a password, and then now you have access to your account at all times. So it just makes sure that the data is safe. If you are a public user and you've uploaded some data and you've done some analysis, it's really hard to go find that public user because it's just a random ID. So it's better to create an account. But right now, if you're following along and you want to just do the test samples, you don't need to make an account. You can kind of just go over to this tab here, uploaded, and you can get started. So now that I'm kind of done talking about that, if you want to make sure that your analyses and data is stored, then you might want to make an account. If not, public user is also fine if you want to just test it out for now. So the overview of Crescent is there's 3 main pages. There's actually only 3 pages here. So all these kind of tabs and stuff are more just pop-ups. We have pop-ups here and pop-ups there. So the main 3 pages are projects, and we have a set of projects and kind of like folders. And then inside of those folders, you have a couple of things happening. We call this the runs page. So in the runs page, it gives you different things like runs and, actually the analyses themselves. We also have the data sets. So kind of like files in a folder, we have data sets inside of a project. And so what actually is happening here is, and it's probably a better idea for me to draw this, but we can have projects, and projects contain a set of data sets. So those files that I mentioned, and then a combination of these data sets makes up a run. So you can choose to organize your project and data set and run however you'd like. It's possible that you add all your data sets for your study in one project, and then maybe you split out your runs into your different conditions. Maybe you want to do this QC separately between treatment and normal and all that kind of stuff. So that's just the kind of general overview of the kind of schema for us. But as a user, you're really just looking at projects, and then once you click into a folder, you're looking at runs. Clicking into a run, this is the 3rd and final page, which is visualizations. So visualizations, and this is where you will get all of your results for your run. So with that said, projects page gives you a lot of information. So there's a couple things happening here. So we have this tab called public. These are all the published data sets. So these are all curated by the Crescent team. So you can notice that it's a curated data set because it has this Cress ID, and if you click it, you'll have this Cress ID. Your data will never be public unless you want it to be. So if you upload your data, it doesn't magically become public or something. There's an actual process to get that data curated. We have to make sure that it's linking it to a paper and making sure that it's available in other sources like what's that called, the gene expression portal and stuff like that. So making sure that that data is actually open source and available, not just through Crescent. So as a user, you have full control over your data. The uploaded data never becomes public unless you ask us to make it public. So over here, we have these published data sets. These cards kind of give you different information about, what are the runs that have been completed. Maybe you have some failed runs, maybe you have some pending runs. There is some information about who created the project, the data was created, the number of data sets. So again, how many files are in that folder. Hovering over this tab also tells you that information, gives you the details. Over here, the author has delineated a single cell map of diverse immune phenotypes in the breast tumor microenvironment, right? So breast cancer cell lines. So that gives you some kind of information as well. And also, sometimes there will be, oncotree tissue types and tagging in order for us to filter by, maybe we want only the cancer, or maybe we want only the non-cancer, or maybe we're looking for a very specific tissue type, for example, brain or breast cancer or whatever, right? So this will give you some extra kind of filtering opportunities as well if you need to look for data. And of course, the same filters apply in your uploaded. So the uploaded and public look the same, except that the public is public, right? So inside of the uploaded tab is where you bring your own scRNA-seq data. So I'm just going to quickly point you guys to some data that you can use right now. So if you scroll over to the bottom here, so if you want, you can toggle this non-cancer or not, but this PBMCs from 10x mixed chemistry, these are just blood cells from 10x that are provided, and so these are just free. You can download them from 10x directly, but it's actually easier for us to just download it straight from Crescent. So what we want to do when we're downloading data sets is to just click this little cloud icon. And what it'll do is it'll download, should be pretty quick because it's only 1,000 cells. And that'll give you some test data to get started. And it should unzip and you should be able to see a folder with that data. And so the way that this data is structured is in a gene by cell matrix. So I'm just going to go over to the docs here. So we have genes and cells or barcodes and the counts of those, the mRNA counts of those cells at a particular gene. This format is still acceptable as a, you can still pass in gene by cell, gene by barcode text files. Sometimes, different methods only output this data, but it's highly inefficient because there's a whole bunch of zeros and empty kind of data that comes with single cell RNA sequencing. So it's better to just compress all of this into what we call an MTX format. And so there are tools that will automatically do this for you. There are tools that Crescent provides that you can do to convert these. But usually you can just output, these are the outputs that you get directly from your sequencing center or, directly from that cell ranger mapping tool that I was talking about, particularly for 10x. And so what that file is doing, let me just quickly change my screen share here. Let me know if you can't see my folders here. So what that gives you is this this matrix basically split into 3 files. So you have this barcodes file, a features file. So essentially we just strip out the rows, strip out the columns, and then we have this mapped matrix that has removed all the zeros. So we don't have to worry about how this file is being created. It's kind of automatically generated from Cell Ranger. Cell Ranger provides a lot of documentation on how all this is happening via splice or aligners, but for our purposes, we should be given this data. At least we should have access to the Cell Ranger outputs coming from the sequencing center. So once we have that, what we can do is, so all I've done is kind of just clicked download on this PBMCs from 10x Mixed Chemistries. It just gives you an easy click to get the data. And so now let's just go ahead and upload our first data set. So again, public won't give you access to this plus button because you can't add anything to public, but inside of uploaded data, you can have access to this big plus button that will create a project. So creating a project is pretty straightforward. You pretty much just say, what is this project? What is this project? And maybe a short description. And so the next thing that you want to select is, okay, you have your details, you're going to need some data. So there's a couple options here. So I did mention that, Crescent provides a whole bunch of data sets that you can combine with yours, with your own data. So you can select from these projects and kind of, build out a larger data set. Alternatively, you can select your own projects and you can also upload data. So with uploaded data, it's just a folder of all these MTX files. And the one that we just downloaded and unzipped, we can just grab it and plop it here. And so what you should get is a quick validity check to make sure that those files are valid. And once that's done, we should see that data populated here. We should. Let's see if this is bugging out right now. All right, so there could be something wrong with the upload here, but I should figure that out by the end of this talk. So alternative to uploading your data. So this is probably just a bug because I did refresh the server this morning, so it could have been something with that. But we have another way of selecting data. So this isn't the end of our road here. So what we can do is from our public data, we're going to go ahead and select this project. So the upload, if you follow the documentation later, you can kind of see how data is being uploaded. It's probably just something to do with the server right now, but I will get that fixed. So selecting this project should give you this data as well. So if you head over to create projects, we have this selected in blue, we can head over to create project. You'll create the project, and what will happen is you'll have now access to this external data set or this public data set. So what should have happened is you should have seen your upload. Maybe you guys are seeing it. I might have like a cache issue or something, but essentially, even if you are not able to upload data, that obviously is one of the core functions, so that will be fixed. But you should still have access to data here. So again, within this folder, we have a certain number of files available to us. So if we were to click this, we can see that there's all these data sets available to us. So if we go back to our project here, so one thing I do want to mention as well, using the back arrow is going to screw things up. So we have our own little back arrow because it's all within one single page or 3 pages. So, it might get confusing because you might use the back arrow to go back, but it's not going to be helpful. So there is a back arrow there, but essentially now that we're inside of our project, we can now do a bunch of things. So our project or our runs page has a lot of, or within our folder or runs page has a lot of options here. So we can choose to share this project if we want to do collaborative analysis. All you need is the email that the person signed up for Crescent with. So if they're on Crescent, it will add automatically. If not, then it might not show up there because they're not a user. So if I add in my name or my email, you need the exact email. So obviously you can't just kind of search for people who are in the app. If you have the exact email, it should connect you to that user. You also have an option to delete that project, obviously for reasons, maybe some errors in uploading. That just happened. So from here, we can, now that we have some data in this folder, now we can start to take a subset of that data and create a run. So for our purposes in this initial run, I'm going to just run the very first data set. So, or not the very first data, only one data set. So I'm just going to say test PBMCs 1K, and I'm going to select as many data sets that I want. In this case, I'm just going to run through an example of selecting one single data set. This one here, 1K PBMCs V3 Chem 3P, so 3 prime, V3 chemistry, and 1,000 PBMCs. So once I've selected my data set, what we're doing now is we're saying, okay, well, I have this project with a bunch of files. Now I need to create a pipeline or a run with a set of these files. And so I'm giving that a name and selecting it. So once I do that, I can create run. And now I'm given a bunch of options to set all of my parameters for my pipeline. So again, you can kind of see the general 9 steps, or in this case, it's not the full 9, but there are some kind of hidden away in annotations after we're done our pipeline. But now we can start to go through the different steps of the single cell RNA-seq analysis. So you can choose to use the buttons for QC, because sometimes you might have to QC like maybe hundreds of data sets. So you can choose to download this, and what you'll get is the CSV file. And when you open that CSV file, you can just edit the parameters for each data set. Maybe you want all of them to be the same or something like that. And then you can re-upload that. And what that will do is we'll change kind of the configuration here, but we're just going to use the front end for now. So sometimes you'll bring in data that's already been QC'd and already been normalized. Then you probably want to select no for this option. But for now, we're just bringing in MTX files that we need to run, and we're going to apply our own QC filters. So each one of these kind of boxes is a different set of Seurat, single cell RNA-seq analysis tools, except that when you're running this analysis in R, and if you did want to go and see, what exactly we're running, I'll provide all the scripts and all the kind of open source, workflows. But we've kind of made it simple for everyone to just go in and kind of just hit submit if you really wanted to. But if you wanted to maybe make some adjustments to your QC, for example, the max mitochondrial fraction would show you that you don't want 20% of mitochondrial DNA, which greater than 20%, and that might tell you that these cells are dying or not high quality. the actual number of genes and number of counts that will tell you whether there's doublets or so few genes like an ambient RNA and so on and so forth, just floating around. So these type of filters are kind of preset for you. So you don't really have to do anything, but if there is something specific about your data that you want to bring in, this is where you would do that. Essentially each one of these maps to a Surratt parameter. I'm not going to go through each one like that, but these are the general kind of parameters that you would do for QC. You can also choose to downsample as well, but that's assuming if you have greater than 1,000 or 5,000 cells. The next step that we need to do, so once we've set, as you click through and change these are automatically saved. So moving on to the next step, we can see, this got saved at 0.3, and we can just keep moving. Normalization. So there's different types of normalization methods. So you can choose to skip that normalization if you're bringing in, RPKM data or already pre-normalized data, but choosing between these two different Surratt methods. Log normalize is just very simple. It's dividing by the total counts for that cell, and then a scale factor, just a very simple normalization. SC transform is a little bit more invested. You have to do, it's a very specific, statistical kind of normalization method that Satija Lab and Surratt has come up with in order to do integration analyses. And I'll talk about integration analysis in a second. But in this case, the default is always SC transform because we also provide integration analyses, which I'll talk about. So in this case, we don't have integration. And what I mean by integration is the integration of multiple different single cell RNA-seq data sets. And that can be different type of modalities, different, conditions. It could be even different species. So we don't have that example right here because we've only selected one data set inside of our run. And so you can see that when I hover over the name here, it tells me what my data sets are, or what are the files that I chose in order to make this run. Dimension reduction. So the default is 10, but 10 to 15 is usually a good number for high dimensional data. After 15, you kind of don't really get the features of that data or of single cell RNA-seq data. You kind of lose some of those features. There are a bunch of plots that Crescent gives you from Surratt in order to kind of fine tune these, but the default 10 is usually pretty good. Cell clustering. So this is clustering resolution. So if you want to obtain a larger number of cell clusters, so as you increase this, every cell could potentially be a cluster, right? Because every cell could have something that identifies itself differently. So this is a little bit of a play as you go, or kind of something that you would adjust between different runs just to find the perfect clustering resolution. So this is just to find that cell's nearest neighbors and kind of make sure that the clustering is somewhat accurate. And then the differential gene expression, what's the p-value that you want to look for, the return threshold, do you only want significant or up to a specific threshold of significance for your differentially expressed genes. Then the last menu here for our single data set run is saving data. So sometimes the unfiltered matrix files you already have access to because you can just click the little download icon. But if you want these in your download, in the final download, you can select yes. If you want the QC filtered matrix files, you can select those as well. And finally, the one that's probably the most important is the R object. If you want to take this and do further analyses, which a lot of lab members for us, the use cases use Crescent for an initial exploration and then pull out the R data and really go deeper into the analysis with Surratt. So if that's something that interests you, you can include the R object. If the file is super huge, you might have to contact us just in case the R data is just not able to be sent, and we'll figure out a way to get it to you. But for the most part, you can download all of your analyses, and all of that is kind of open sourced. So now that we've set all of our parameters, the last thing that we have to do is submit run. So If we click submit run, it'll tell you to refresh the page. And as you refresh the page, what you'll see is, a couple of more tabs that have been unlocked. So pipeline is kind of now locked. All this stuff is submitted. We can see logs. The logs page is kind of running. So if you were to go see what's happening, it goes through each step and tells you live as it's running inside of that, workflow execution. So now the job is running. We're normalizing with SC transform, and all of this stuff will go really fast for this 1,000 cell data set. The 1K cells will run really quickly. I think within a couple of minutes. We'll get stuck at differential gene expression because that is a high RAM requirement, and it's going to take some time. But other than that, all the other steps should run pretty quickly. Of course, if you scale to the level of like hundreds of thousands of cells, you should expect, maybe a couple of hours of runtime just to run this inside of our high performance compute. So other options that you have. So once you've submitted, you can also cancel this pipeline. Let's say you ran the wrong parameters, or for some reason, you can cancel this. And as results come, they'll be populated in this results page. So right now, we might have some QC results and stuff, but we can wait till the end of this because it's going to be really fast. So, okay. So while this is running, and a couple of seconds here, did I lose anyone here with these with this walkthrough? Because I know I'm going pretty fast, but for the most part, the idea around Crescent is for it to be intuitive. So it really is single cell analysis in a couple of clicks. As you enter the data, or as you enter the app itself, it should only be 2 clicks to get to some sort of meaningful visualization. Getting your data to run should be a simple upload. I'll figure out what happened there, but should be a simple upload, and then setting a few parameters if you choose, but you don't always need to set the parameters. What we've done is we've run a bunch of benchmark studies in order to actually kind of set what are the best parameters, or what we've seen as the best parameters over the years. So I can go back to my data set here. Or my project here, and it looks like that run is completed. I can see that it's green, and I can click here and I can see a few details inside of my run card. So obviously this green means that it's done. Yellow means that it's submitted, and pending means that you haven't actually started to run it yet, and red obviously failed. So it tells me, who created it, what time, how long it took. So this took a minute to run. It was one single data set and 1,100 cells. So if we click into this run, now we're taken to our visualizations page, and we can actually start to explore this data. So some of the results that you might get here are QC, TISNE, UMAP, gene expression violence, and dot plots. So QC is one of the first things that you're probably going to check. You'll notice that I'm also adding different tabs. So if you want to if you want to add different visualizations, you can do that here. And you can also compare them over here. But we're just going to look at one for now. We're going to look at QC. You can also save these plots. So in case you are looking for a very specific gene and you wanted to kind of save that visualization, you can do that as well. So you can kind of see here what's happening. So with our number of genes, it kind of passed all the filters, our before and after. We have a couple of stats about what was actually removed, or which step removed what. So you can kind of see here, we didn't remove any cells based on number of genes or number of reads. So maybe we didn't have any doublets or ambient RNA, but it does look like we had some dead or dying cells, because 87 of those cells were removed. And you can see here, some of these cells had 90% mitochondrial gene, right? So most of these were mitochondrial genes, and that just shows that this cell was probably in a state that would be considered low quality, i.e. necrotic tissue, or in a dying state. So we've filtered out these cells, so you can kind of right off the bat see, okay, our QC filtered out some low quality cells. And we can also filter, eventually when we do our integration, which hopefully I can do in 5 minutes, do justice in 5 minutes. We can also check a bunch of other QC stats. So you're able to see some numeric data, like, for example, the number of genes. So obviously this is taking advantage of that UMAP filtering that's being done at the end of the pipeline, but it kind of tells you, hey, maybe this cluster, this cluster that has a really high mitochondrial count, maybe there's something special about it, something that you want to, know, something here interesting is happening with, a high level of UMI counts. So all these things are, useful to some degree for depending on what you're doing. So that's kind of what is the, bring to Crescent part of this is you need to be able to kind of know which genes you're looking for, what interpretation or what you're trying to get out of this data. Crescent makes all these things easy and explorable, but the onus is kind of on the user to actually know what's important and what's kind of being pulled out. So in this case, if any of these counts are important to you, or you bring your own, numeric metadata, then it might be pretty effective to use this gradient just to visualize that. So that was just the QC menu. So there's also our actual exploration and gene expression itself. So TISNE, nowadays we're using UMAP. The difference is, the distance in UMAP is actually a measurement. So the distance actually matters here, whereas in TISNE, it does not matter. So both equally effective of showing clusters and summarizing high dimensional data into a visualization, but kind of the default nowadays is UMAP. So with UMAP, we kind of see all these different clusters, and what's happening here is the pipeline has determined that each of these clusters are a different cell type, or based on your resolution, it's saying what actually makes these cells different, right? And so you can kind of see the separation between three types of cells. It could be, or three types of, three groups of clusters. And so that could be an indication that, these groups are different within each other. These could be different types of immune cells or blood cells because these are PBMCs. But within that group of, immune cells, for example, we can see this separation of function and gene expression. So let's say we had our favorite gene. We're looking for a gene when we're exploring this data. What we can do is we can take a look at these top differentially expressed genes. So these are the genes that are the most different between every cluster. So with cluster 0, these are the most significantly different genes. We can see the average log fold change and p-value as we hover over. And so clicking one of these gene expression buttons or these genes tells you what the actual normalized gene expression is. And so this kind of gives you an idea about the different clustering and also the organization of this data, but also what actually is happening in this data. So, how can we maybe explore, BRCA or BRCA mutations, or can we explore, TP53 and see which clusters actually express these things? So the gene expression buttons are kind of an easy way for you to handle that. So there's also another way that we can do gene expression. We can use this gene expression violin. So I'm just going to make sure I pick the right gene here. So I'm going to pick this one. We can see that it's kind of highly expressed in this pink and red cluster. We can head over here and pick the same one. And what we'll see here, if we kind of collapse these plots over here, is we can see a better representation with the violin plots to see, the normalized expression of this particular gene. So, when you're looking at a cluster like this with all the colors and stuff, it might be hard to read. So you might want to kind of separate it out into the gene expression violin. So, the last plot here is a dot plot. So with the dot plot, similar to the gene expression violin, you need to select the gene, the size and color determine specific things. I always get these mixed up, but the size of the dot refers to the percentage of cells within a cluster that express a specific gene. And then the opacity refers to the average expression. So the size refers to how many cells, and the opacity is the expression of those cells for this particular gene. And we can add other genes as well, and you can see the dot plot kind of take shape. And there are, obviously ways to compare these as well with our other plots. Okay, so I'm out of time, but I do have a couple of things before. So I did show you guys one data set, but Crescent also provides an integration analysis. So integration analysis from Surat is just a way to anchor your data sets, most similar data sets. So this is coming directly from Surat. Hopefully you can see that. So in a way, you can combine different modalities of data, different experiments, different species, even like mouse and human. And what happens is, when you're anchoring these data sets, the similar expressions will anchor together. But as these unique clusters are being mapped, they're probably not going to be anchored, or they're going to be anchored randomly, as you can see. So that kind of identifies that cluster as being different. And during the integration analysis, more focus is put on that during the clustering. So I'm not going to go through that, but essentially the way that you would do that is by selecting more than one data set. And that's all you have to do that. Selecting more than one data set gives you options in the integration panel, which are all explained, and all of these defaults can kind of help you. But the only thing you really have to do is select the data sets that you want to anchor by. Usually these data sets are the most different data sets. So maybe conditions and stuff like that you select, and then all of your anchored and integrated data should show up something like this. So with 5 different data sets, what you'll have is abilities to look at each data set separately. You'll look at UMaps and be able to kind of select, maybe I just want to look at, not that one here, maybe I just want to look at one single data set, and so on and so forth. So you can kind of filter through each data set as well, but also see the total kind of integration. And the reason you would do something like that is so you could take the reference data sets of, maybe you're bringing in some immune samples, pull out all the immune cells, and maybe tell you what's in the tumor microenvironment that's not immune, right? And might be interesting to you. So there is also GSVA and all this other stuff that we can do, but I'm not going to go into that amount of time here. But there are, a lot of documentation on this. I'm available on Slack if you need to message or anything like that. I will fix that upload. That should have worked. That's very embarrassing, but it did work. I think I reset the server this morning, so something may have screwed up there. But quickly before I shut up, summary: Crescent is a web application to analyze, integrate single cell data. It's free to use, it's open source, no dependencies. It uses high performance and publicly available cloud and commercial cloud. Users can collaborate. We have multi-data set integration. We have gene level expression querying and cluster labeling, as well as cell label. Metadata visualization, which I didn't get to show, but it's quite easy. You just drop in data. It's all pointed out in the documentation itself. And the last part here is just the resources. So if you want to check out any of the docs, it's in the info. If you want to check out any of the scripts, it's in our GitHub, the code base, the publication, Slack, and how you can contact us. Sorry, went 3 minutes over.
I don't want to keep them. Okay. So do we have any questions? I think we seriously run out of time. Okay, I just wanted to end by saying that, so this is like a test run for us, for Panoramics, but we really wanted to do this for the biologists, because we want more biologists and bench scientists to be on board. So there were some things that Sulaksan brought up, and also Paul and Emily brought up last week, is that, for instance, mitochondria is the de facto parameter that we use to identify dead cells, right? And integration is one tool in which we cluster, or we actually overlay different patient-derived samples. But I'm a cancer biologist, and he knows as well that integration doesn't work well for cancer types. Yeah. And at times like this, we need the biologists to step in. So this is like, these are tools which we want to encourage everyone to use, so that you don't have to feel left out. It's not only the domain for computational biologists or scientists. And we need expertise to come in and to join us, to understand, to look at a UMAP and to tell us that, or that's kind of like a wrong cell type labeling, if your clustering is not differentiating or it's not, highlighting. And that always happens. Yeah, that always happens, right? So this is like a set of, tutorials to help that. Yeah, but I think I didn't kind of send that message across too well, but I'll improve the next time. But this is the real reason. And there are all these tools as well. Second, there are all these tools that people don't know about. So we want to encourage everyone to use Kobe and Crescent and also CBioportal. And we'll add on to these tools. So that's all I have to say. This election.
Yeah.
Yeah.
Thank you. Yeah, definitely the interpretation is it's not clear cut. Crescent's not going to tell you exactly what's in your data set. It all depends on your parameters. I think one of the early things with this toolkit was no one could decide what tool to run or what parameter to choose. So essentially we're just trying to standardize that, but that's not, a one, it's not a fit all solution, right? So there's obviously these custom things that you have to do yourself, but Crescent is supposed to be that initial exploration so that you can dive into the data without a whole maybe PhD of learning how to do single cell in R, but with a couple of clicks and kind of cute buttons and visualizations. I think
I shouldn't have to learn, right? They are coming with their own expertise.
Exactly.
Right?
Yeah.
So we need to leverage on that.
Exactly.
So yeah, so I think we ended. So we are ending now for Crescent.
Yep.
We will be starting CBioportal. Do you have any questions?
I had two general questions. One is, can you at the end get the publication ready version of the workflow? So that, if you use the tool, then you can go to your publication, say, this is how I did it.
So let me show you that. Yeah, so over here, when you download this, the run that we did, you can see there's a file that basically gives you everything that we've ran. So this download results, if you open it up, it should be a big zip file. Sometimes if you have the R data, it might be too big, but normally it's within a gig or something like that. So opening this file kind of gives you all of the output files that are coming from Surat. Most of these are, for example, the actual TSVs of the data itself, but you can see, there are all the kind of publication plots. Usually if you want to do your own custom stuff, you would take the object and do it. But for the actual, yeah, so exactly, yeah. So we include the script that we ran in order to actually get all this data out. So if you, in the download itself, you get this R file, and it basically tells you, where is this coming from, the resources, what did we run? And you also have parameters that you've passed into this. So that's how we did this whole workflow management part. So all this stuff is supposed to be like, you grab it and you just go to a publication. Sometimes you want to maybe, change some of the colors and fonts and stuff. That's when you would use the R data object. But for the most part, if you want the default kind of plots, all that is coming from Surat as well, and Crescent, obviously. Yeah.
And then one sort of very specific question is, are there runs sort of like batches, or are batches managed in a different way? You mentioned that you can control for batches for the
what do you mean by batches? Like between different technical variants? Yeah, so in this case, the runs are a collection of data sets. So there's just a collection of samples. So the batch correction is kind of just done through the integration. So SC transform kind of handles that for us through Surat. The anchoring itself is supposed to handle that between modalities and species and stuff. with one single data set. The only thing you can do is normalize across those different cells for size and stuff like that. But for the most part, batch correction usually between different batches, like you said. Right? So in this case, that one single data set is probably one batch, like one experiment is usually how it's grouped. But you can choose to split that up however you want as well. If that's something like maybe you have 2 different treatments, and even though they were run at the same time, maybe you do want to do a batch correction anyway, right?
General question is, you know, the name Crescent is referring to cancer, but it's not just for cancer.
Exactly.
Right?
Yeah. No, that is a good point. So, yeah. So all of our reference data, which you can eventually anchor with, is all a bunch of cancer data. There will be normal cells, a lot more normal cells in the future, but yeah, the idea is mostly coming from, you know, just cancer single cell analysis, and all these parameters have been tuned for cancer, but again, you could bring in any normal cell or any new set of data that you want to analyze, and it could be maybe a new cell type or something like that, and everything would work the same. You just might have to tweak the parameters a little bit more specific to your data. But yeah, it's the cancer part is more in the infer CNV. So if you want to see the copy number between cells, that part I didn't really get to. I think if we do this next time, we probably need to do like 2 hours because it's like I realize there's like, this is the intro for sure, but there's a lot more to this that are kind of experimental. Like we can do these annotations on top. So I didn't talk about this one really important thing, but custom metadata, you can drag and drop your own custom metadata so that when you go to look at your results, they're labeled as you labeled them, right? So maybe like B cell receptors, T cell receptors, those are measurements that you can get orthogonally, right? So those things might be effective in trying to delineate which cells, what cell types and stuff. But yeah, that's kind of beyond the scope of this intro, but definitely maybe we could do, yeah, sorry. And okay, so do I end the call? Okay, all right, bye guys.
So thank you, everyone.
Sorry.